Alright, so now we are going to talk about the sequence part of the sequence analysis. And this is one of the biggest application of bioinformatics, because in bioinformatics we are dealing with information, particularly sequences. What sequence? Nucleotide sequence as well as amino acid sequences. So sequence analysis is very important thing. Okay. So the sequence that we get, there are primary sequence data, okay, and there are also secondary sequence data. Both are available. The primary sequence data, we have talked about what is primary data. That is a data which is available in the general sequence like Genbank sequence and all, Genbank data. And secondary data are specific data, okay, not generalized data, specific data. Both are equally important. And sequence formats are the formats at which the different sequences are available. So we call it sequence file format. So it's different kinds of file, just like image is a type of file, video is a type of file, audio is a type of file. So in this case also we have different types of files, sequence file and molecular file. We have sequence file format and molecular file format. Okay. The sequence file format represents what? DNA sequences, also represents protein sequences. Okay. So among this sequence file format, there are some examples that we will be discussing in a moment. There are few examples, and there are this molecular file format. They are little different. Molecular file format, mostly they composed of 3D structure of protein, 3D structures of protein, and basically the data that we obtain is obtained from X-ray crystallography. Okay, from X-ray crystallography data directly we can feed in, or we can go for NMR spectroscopy data. We can feed in and get a 3D structure of proteins, and those data are stored in molecular file formats. We'll see some example of molecular file formats later on. Okay, let's first talk about the sequence file format. Example of sequence file format. These are all examples of sequence file format. So we have raw format, Fasta format, Genbank file format, Gcg format, Gcg-msf format, Embl format, Phylip format, Nexus format, Clustal W format, PIR nbrf format, Uniprot swiss-prot format. So many different kinds. They have their own format. So formatting here is, in case of a video, we know that the file name. So let me write file name, then dot, it can be MP, it can be MP4, file name dot AVI, okay, file name dot MPEG2. So there are different names like that. These are the file formats. Similarly, we have file formats for sequence. For example, if you start with the raw format, if we start here, the raw format is just the sequence of DNA, either DNA sequence or protein sequence, nothing else, just the sequence. Okay, and in this case only IUPAC characters are allowed. There is no digit, no space, no digit, no space. So basically continuous writings like that. Start with A, small a should be there, AT, whatever it is, G. So these are all nucleotide sequences. Okay, these are raw format, no spacing, remember that, and no digits, number digits. Then we have Fasta format, second type. Fasta format contains a one line header. This is a header. Now what this header is composed of? Starts with this symbol, and rest is the name, description of the sequence. FOSB underscore human protein FOSB 338 base pairs. So again in this case blank space is ignored. So we will not add any blank space in a FASTA format. Okay. And FASTA format is the program or format which is accepted for multiple sequence alignment. Okay. And termination of the sequence is marked with an asterisk like this, star mark like this. Okay. Now we have Genbank file format. It's a NCBI database. They have their own file format which is known as flat file. This is divided into three different sections: header, features, and sequence. So what header consists of? Description of the sequence, let me write with a different color, source, organism, length, scientific name, and biological significance. Taxonomy, related information, all these things are present in the header. Okay. It also carries transcriptional units, information regarding transcriptional units. Also carries information regarding mutations. So it's kind of all-in-one information. And bibliographic references. And then there is a rest part is the sequence part represents the composition of the sequence by IUPAC symbols. Okay, so these three components are of flat file format. So there are different headers of the field and there are description to it. For example, locus. So short name for the sequence is mentioned there. Definition means description of entry. So there are different headers and mentioned. So I'm not going to say individual out of it. This is flat file format. But remember, flat file is format of NCBI's own file format for NCBI's own databases. Then we have, I think this is the fourth one, GCG file format. So this is known as GCG means what? Genetic computer group. Genetic computer group format, GCG. Okay. So in this format what happens? It begins with annotation. So this is the line, whatever sequence version is mentioned for the nucleic acid. In the bracket they will write either for the nucleic acid or for the protein. If this is Na, then nucleic acid, and if it's written as NP, then means AA, if it is a AA, so AA underscore whatever value, then it is for protein. A for protein, Na for nucleic acid. Okay. And the next line contains informative text following ... at the end. So in this case, this ... the dotted lines, this is by choice, not just a dotted line. Okay, so this is the sequence number starting from 1 and this is 51. Dotted line is the ending. So test sequence length is given 4303, date April 10, 1997, time, type, everything is listed here. Okay. Then we have another file format, Gcg-msf. Gcg was computer, genetic computer group. But when the genetic computer group file have multiple sequence file in it, then it will appear as a MSF format. Multiple sequence file. Multiple sequence file. Okay. So what is the file format? So it will be something like NA underscore whatever value, and then finally at the end it will be the version, or it can be AA, whatever it is. This is for DNA and this is for protein. Okay, but there is one more catch, and in this file format the first line is mandatory and it must have MSF written there. MSF. Also, this first line indicates the sequence length, type and date. And after that, the next line is mandatorily kept blank. Okay. So start with MSF, first line, next line, blank. Then the sequence, and initially the first line that contains started with MSF, that includes the sequence length, type and date information. So the third line is for the sequence separately, and this follows the rule of GCG format. The sequence follows the rule of GCG format. Okay, now we'll move to the sixth, EMBL type. EMBL, this is followed by European Molecular Biology Laboratory, EMBL, for all the database under the EMBL. So they have different types of lines and it contains all the records. Each entry begins with an identification number or ID line. It starts with an ID line and then termination is with double slash. Remember that. Start with ID, then termination with double slash. I'm not showing an example here because when we do the practical you can see the details and example, but remember one thing, start with an ID line, ends with a double slash. There are multiple lines will be following through this. Okay, from the ID line to all these different lines, XX, SC, SB, DT, all these different lines are there and they have different roles to play. They have different description. Individual have different description. It's not that difficult to understand the file format for EMBL particularly because there are so many different lines and each line have different information. For example, after ID there is a XX line. So this is used in the place of blank lines. Then we have AC, a line with AC segment known as accession number data. Then SV segment contains nucleotide sequence identifier in this section. So there are multiple such sections. It's not feasible and scientific to try to mug it up. You don't need it. Okay, so you can, if you go to EMBL database and you try to work with some sequence, just select individual lines and take the cursor there and it will automatically show up the features of those lines and formats. We have Phylip format. This is by molecular phylogeny software, particularly by phylogeny software. What kind of phylogeny? Molecular. By molecular phylogeny software. And there are two formats used here. Interleaved format and sequential format. Interleaved and sequential format, both are used. Let's take a color. Interleaved and sequential formats are used. This particular, you can see this starts with two numbers, then the sequence line comes in and at the end we have dotted lines. Okay, so first number represents the number of sequence in the file and the second number is the total character present in each sequence. This is number of sequence and this is total number of characters in each sequence. The next line onwards, the sequence itself is displayed. Okay. So the way we represent the sequence, by a sequence title which is not more than 10 characters. 1, 2, 3, 4, 5, 6, 7, 8, 9. You can see 9 characters in both. Okay. Sequence character line not more than 10 characters, then rest of the sequence will be provided. We have Nexus as a file format. So Nexus is a file format if you retrieve the data via POP and MacClade program. POP and MacClade program. They use Nexus file format. So basically this POP and MacClade, these software programs, they use Nexus file format. Starts with a number, Nexus. Start with this data. Nexus is the term that they started with. Okay, and then the beginning of the data, and then what you can see, the dimensions of character is also told 60 n character, format data type DNA, gap missing. Gap is denoted with dash, missing is denoted with question mark. So basically what they mean is already stated at the top. Quite easy, quite simple, straightforward. The information regarding the dimension, the character, format type, whether it's a DNA or protein data. If gap is present, then it's a dash, missing is question mark, is already stated. So what else we have? We have matrix here. Cow, chicken, mouse, human, rat, and for them the data is provided. That is actually Nexus. Nexus is a data not needed for a general purpose. It's only needed if you are working with MacClade or POP softwares. Then comes Clustal W format. Clustal W, there are different versions. The version is 1.82 mentioned in here. Clustal W is another format which we can use for multiple sequence alignment or MSA. There are two formats that we predominantly use for multiple sequence alignments. One is the Fasta format, another one is the Clustal W format. In Clustal W format, the most recent version may be changing from this value that whenever you are seeing this early recorded version here. So you can see what is this format. Clustal W and Clustal X, there are two formats. They start with Clustal, the file start with Clustal, all caps, and the version that we use, in this case Clustal W 1.8, that is the version we are using. And then the alignment is written in blocks of 60 residues. You can see the alignment data for mouse and human, LEP mouse and LEP human, the data is already mentioned in here. Every block starts with the sequence name. The sequence name is in this case LEP mouse. The second sequence name is LEP human and the sequence is mentioned. See that. Okay. The input sequence are obtained and a count of total number of residues is shown at the end of the line. Total number of residues will be shown at the end of the line. You can see here at the end of the line what we have stars, 4 star, 1 dot, star, this and 2 stars. So what they mean? This means residue in that column are identical in all sequence. As you can see, identical, 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 different. So different. Semi-conserved sequences, a dot. Conserved substitution is this. Okay. So these three places are different. So that is already mentioned in here. Star means residues in the column are identical to all sequence or conserved sequence, and this means semi-conserved sequence substitution. Means the conserved nature is there. Semi-conserved means not totally conserved, semi-conserved. Maybe due to some sort of substitution, the conservedness is not 100% semi-conserved. Then what we have? PIR nbrf format. That is again from National Biomedical Research Foundation. National Biomedical Research Foundation NBRF format, PIR format, which is very similar to that of the Fasta format, although Fasta is widely used. It also starts with this sign like Fasta, this symbol, followed by the sequence type code. In this case, this is sequence type code CRAB ANAPL type code. Then so this is type code P1. This is the first type code P1, and then semicolon, and then the next line sequence name and description, alpha crystalline B chain, and the description is 200 bases. So the protein chain here is alpha crystalline B, and there are 200 bases present there. It's an amino acid sequence that we are looking at. So sequence means obviously either it can be amino acid or nucleotide, in this case amino acid sequence. Then the sequence is followed from the following line, and once the sequence is end, star is placed. So whenever there is a star, we can tell yes, the sequence is end. It's very important to mark the start and the end. So we'll mark start with this, we'll mark end with the star. Okay, very similar to that of the Fasta format. And what else we have? Uniprot, swiss-prot format. So basically Uniprot, swiss-prot, these are protein databases, isn't it? These are protein databases. So there are annotated protein databases, and their entry is composed of lines. For the standardization purpose, the Uniprot or swiss-prot, if you look at as closely as possible to that of the Embl format, very similar to that of the Embl format, similarity is there, but little difference is also there. For example, in this case there is a line called GN line. Gene name contains the name of the gene, and also if it's for the, obviously it's for the protein, so name of the gene which actually translated to the protein. And there is also another line is OG line, organelle. GN means gene line, this is gene line, OG is organelle, indicates the location or origination of the gene, that is organelle. Okay, so both informations are added with the Embl type of format, similar format, but the thing is gene line, name of the gene for the coding of the protein is mentioned, and then location, origin of the gene, origin of the gene is already told with organelle or OG sequence. Okay.